Hi everyone, this is Sadia Jalisi, and today I'm going to explain a presentation that I made about a research article named Gut Microbiota's Causal Effect on the Risk of Periodontitis using a two sample Mendelian randomization study. First of all, I will tell you why I chose this study. Actually, I am a dentist by training and I'm very interested in microbiology related to oral health and dental problems. So here, as you can see, it's related to periodontitis. That means periodontal problems and the gut microbiota's causal effects. So first of all, I will tell you about the periodontitis. We can divide this term. First is perio, that means around, and dont means tooth, and itis means inflammation. So it's the simply the inflammation of the gums that are present around our tooth. So the periodontal effects includes the periodontal diseases are the most common cause of the tooth loss. Okay, and periodontitis actually is the most prevalent disease in the world, and in the worst case scenario, it can lead to the tooth loss. Okay, so the it is associated with systemic health problems like heart diseases, chronic lower respiratory disorders, and diabetes. And the risk factors includes the smoking, alcohol consumption, poor oral hygiene. Here you can see the periodontitis and the healthy tooth comparison. The periodontitis is the inflammation of the gums. You can see the red gums, and so the tooth became loose with the gums, and it will cause the tooth loss. Also, you can see the bone loss associated with the periodontitis. That's why is the most common reason for the tooth loss. Okay, the question that can be asked in this research article are: can gut bacteria affect gum diseases or periodontitis being connected to the oral cavity? And the second one is gut microbes can be seen to be involved with many pathological issues such as heart diseases, autoimmune diseases, and cancers. Can it also cause or affect the periodontitis? And here you can see a link between in this diagram that the oral cavity is connected to the stomach, that is gut. So there could be a relation between the gut microbiomes with the oral diseases. So the bottom line answer to the question is the researchers use MR analysis, that is Mendelian randomization analysis, and GWAS, that is genome wide association studies, to answer these questions. They found a definite causal relation of the gut taxa and the periodontitis, and also have expanded the taxa that causes the periodontal diseases. So what's the need of this question? So this is an issue that is affecting millions of people, and it is one of the biggest reason for the tooth loss. And it's having a better understanding and better methods of detecting and predicting periodontal disease. It's very important. So broader implication is to develop more comprehensive treatment for the gum diseases. Now, the gut microbiota, the gut microbiota includes the bacteria, fungi, viruses, archaea, and other organisms, and they have a huge impact on the human health and diseases like immune system, and they can helps in promoting innate and adaptive immunity, and also it helps in digestion of food and cellulose and synthesizing essential vitamins and metabolism of like food and metabolizing biles, and also they can cause cancers because They cause inflammation and it is a high risk factor for the cancer. So, the risk factor in the gut microbiota includes the diet, antibiotic, age, diabetes, and so many others. The Mendelian randomization study that they used in this research is the like it's a type of study that is used in epidemiology, and it helps in evaluation of causal relationship between the exposure and the disease outcome. So it uses the genetic variants like SNPs, that means single nucleotide polymorphism. So as the instrument variables, and also I found out that it avoids some of the limitations that we see in the observational epidemiology. That, for example, reverse causality and confounders. Here is a diagram that shows like three assumptions for Mendelian randomization. And here you can see the first one is A, that shows that the genetic variants is they are should be associated with the exposure. And here you can see the genetic variants should not associate with the confounders. And here in this third one, you can see here the third assumption is that the genetic variants should influence the outcome only through the exposure. So, confounders are not a good thing because they introduce error or bias. So, controlling them is essential to establish a true causal relationship. And these are the assumptions of Mendelian randomization, that the genetic variants should not be connected with the confounders. Okay, so it has these Mendelian randomization has similarities with the randomized control trial. As we know, the randomized control trial are considered the good gold standard because it has the best possibility to establish a relationship between a risk factor and an outcome. So they performed this Mendelian randomization study because, as compared to RCT, they are less effective, and they are very practical and ethical study. And so the Mendelian randomization study is very similar to RCT study because the random segregation of genes separates them independently into exposure and the control type. So the the Mendelian randomization has the same thing. Okay, so they are using two sample MR study, and the two sample MR study is explained over here with this diagram. That they like two sample MR study uses two different samples. As you can see over here, one sample A, sample 1 and sample 2. So they uses two different study samples for the risk factor and outcome to estimate the causal effects of the risk factor on the outcome. So it is helpful when it is difficult to measure both the risk factor and an outcome. At the same time, it also offers the chance to incorporate data from different sources. When the genotype exposure and the genotype outcome data comes from different sources, meta-analysis can estimate the combined effect by using two sample MR study. Okay, so it's an association. MR study. Now we will move on to the methodology. MR analysis. They did MR analysis. Explained before that is they found out the causal effects between 196 gut microbiota taxa and periodontitis, and they considered the genetic variants, that is instrument variables, should associate with exposure only, that I explained, like it should be associated with exposure and not the confounders. So this way they had the strong instrument variables that were established. Now here is a diagram uh, that shows the here it shows the healthy tooth state, and here it shows the periodontitis, and this is a gut, and it shows the relationship of the gut microbiota that causing the periodontitis over here. And these are uh, like they are it's healthy, so they are not causing the periodontitis, and they are inhibiting the inflammation and are inhibiting the periodontitis effect. Okay, so here um, they collected data from the genome wide genotypes and the fecal microbiome data that were connected from 18,340 individuals from different countries, and they used the genome wide association studies. Okay, so it's GWAS is the method of study that helps identify genes that are associated with particular disease. Okay, so they this study like in this study we can study entire sets of DNA of large population, and it's identify small variations in the genome, that's SNPs. Okay, so this study included the 196 known bacterial taxa that were 9 phyla, 16 classes, 20 orders, 32 families, and 119 genera. Okay, so here is about the GWAS, that it helps to identify genes that are associated with particular disease and study entire sets of DNA of a large population, and identify small variation in genome called SNPs, and each study can search of thousands of SNPs at the same time. And here they found out the minor allele frequency that was more than zero point zero 0.05, and SNP wise call filtering that was more than 0.95. Then data analysis was done by WM, um, was done by IVW, that is inverse variance weighted median randomization, and MR. Okay, so all these like here they use these tests, these analysis, and so IVW is the most robust analysis that provided the estimates with heterogeneity. Then they used WM and MR agar, and they proved the validity of IBM estimates. So MR agar regression could be used when there is heterogeneity and diversity in the estimates of IVW analysis. And the WM, that is weighted median, did the same thing like MR agar, and it confirmed the estimation of IVW when there is diversity of heterogeneity. Okay, so each of these analysis has an odd ratio, confidence interval per standard deviation, and p values. And the p value, the two-sided p is less than 0.05, was set as a significant value. And the significant estimate provided by IVW, as I told you before, with the same directional estimates provided by WM and MR agar, was treated as significant estimate. And after the MR analysis, they did the sensitivity analysis and detection of pleiotropy. Okay, here, so leave one out analysis. It's the this analysis was performed to know either the causal estimates were driven by SNPs, and it's a meta-analysis on each subset of the study obtained by leaving out exactly one study. Okay, so the other studies are these MR agar and intercept, Cochrane's Q test, and funnel plots. They all use to detect the pleiotropy. Okay, now we will move on to our results. So here you can see a big diagram over here that is circular, and you can see They did the MR analysis, as I discussed before, like that is Mendelian randomization analysis, and in that they performed three main, that is inverse variance weighted median, and MR estimates. Okay, so um, in this diagram, you can see the results of a Mendelian randomization analysis that was performed to find out the association between the gut microbiota and the risk of periodontitis. Okay, so the it is a circular, so you can see the outer circle and the middle and the inner circle. Okay, over here, and so the outer circle represents the IVW, and that is inverse variance weighted, and the middle one over here, it represents the WM, that is weighted median, and the innermost, like this one over here, you can see, and so it's the MR estimates values. Okay, and also there are the gut microbiota was classified in order, phylum, class, family, and the genus that you can see. Uh, and then it has p-values as well in the middle. And the different colors over here in the circle you can see, like orange, green, white, they show represents the p-values. And the outer on the outer of the circle you can see, like there are so many microbiotas, like 196 gut microbiotas that are classified into or the order, phylum, class, family, and the genus. So it basically shows all the inverse variance weighted estimates and the weighted median and the MR agar estimates. Okay, so what they found out from that out of like 196 bacterial taxa that you can see over here in 196 bacterial taxa, they found out that 11 gut microbiota taxa associated with periodontitis, and also out of 11 they found that 7 microbiota show the positive potential causal effects to the periodontitis, and 2 of them showed negative effects on the periodontitis. Okay, we will discuss is in the further slides as well. So the Provatella and the Lacnospiraceae also positively associated. These are the two microbiota that were positively associated with periodontitis, but there is some exception, like because uh, there the IVW and WM and MR agar all were performed and analyzed by the odd ratio, confidence interval, and the p-value. So all that indicated that positive relationship with periodontal disease, but it's their MR agar analysis was insignificant, but their p-value was more than 0.05, so they were considered positively causal affecting microbiota. Okay, so here let's move on. So here you can see another plot that is showing the Mendelian randomization analysis, and here you can see the 11 gut microbiotas, okay, that are associated with periodontitis. And here the red dots over here on the plot, they show the IVW estimates. Okay, and here you can see the on the odd ratios, that is 95% confidence interval, and the p values are mentioned over here as well about all these bacteria. So and the black bars over here, here they represents the 95% confidence interval. So what they found out from these results is that the seven of the bacterial gut microbiotas were causing increasing effects. So how did they find it? That because all these seven had the over here had more than one odd ratios. Okay, so all these seven had more than one odd ratios, while like enterobacterials, pastorales, that you can see the more than one ratios over here, and bacteriodales as 24.7 group, and Provetella 7, and Lacnospiraceae. Okay, and but the two are exceptions. As you can see here, the gut microbiota that decreases the risk of periodontitis, that are Butyricoccus, and Rumini Clostridium 6. So these two had negative effects because their odd ratio was less than 6, as you can see over here in this plot. So their odd ratio was 0.82, and the Rumini Clostridium has the odd ratio of 0.86, and their p-values was 0.038, and the Lecnes Butyricoccus had the p-value of 0.030. So they did all these analysis over here, and they found out the confidence interval, 95% confidence interval. And the other exceptions, that was the anterior bacterials. This bacteria over here, anterior bacterials, it showed the IVW ratio was 1.37, and the 95% confidence interval was 1.07 to 1.74, and the p-value is 0.012. And the other one, Alice tips, 1.20 odd ratio, and 95% confidence interval is 1.01 to 1.42, and the p-value is 0.034. So these two, Alice tips and enterobacterials, they had like their MR agar was reversed than the other two analyses, but they are not significant because their p-values were more than 0.05. So and so they ignored it, and they included them as and they confirmed that they are increasing the risk of periodontitis. So they are among the seven. And then after that, they perform the sensitive analysis and pleiotropies. Okay, uh, they performed these tests to minimize the bias effect. These tests were performed and showed the following results. Okay, so they performed these. First one is leave one out analysis. Okay, they perform all these tests over the 11 microbiota, okay, that were in this over here, all these 11. They perform these tests on the 11 microbiota to confirm their pleiotropy. Okay, they perform so to minimize the bias effect, and all these prove that the p-value of 11 microbiota is more than 0.05. Okay, so the first one over here is leave one out analysis, and that is no, it showed the no pleiotropy. Then the funnel plots indicated that no bias being caused by symmetrical distribution of SNPs, that is single nucleotide polymorphism. And Cochrane's Q test also proved that there is no heterogeneity, and so MR agar intercept showed that there is a lack of horizontal pleiotropy. So among all these 11 taxa. And so all these sensitive analysis showed that the p-value was more than 0.05 of 11 gut microbiota taxa. Okay, so what would we found out by these results, that there is a very strong relationship, okay, and there is no imperfection between the instrumental variables and exposure, that is gut microbiota. And there is no imperfections, and they are all the results are very perfect by this Mendelian randomization study, and they proved it. Okay, so here you can see the causally initiate periodontitis that was caused by these seven genus Provatella, genus Lachnesperiaceae, Enterobacteriales, Pastorales, and all these seven family Pastoralaceae, family Bacteroideales, genus Alistipus, and genus Esenberglia. And the other two, that is genus Butyriciococcus and genus Clostridium 6, causally reduced the periodontitis, and they were negative. Okay, and then Provatella 7. Provatella 7 was found in abundance in the fecal samples, and we all know that, we as a dentist know that they help in the Provatella gingivalis, and it's a key in activating this bacteria. And we find these bacteria very common in our oral cavity. So, and it causes the periodontitis or periodontal problems. So it's a predictive biomarker and target for efficient treatment and prevention in the periodontitis. Okay, and so the other is genus Lachnespiraceae, UCG008. It also showed the increases the risk of periodontitis and cause inflammation by producing inflammatory factors like interleukin 6, tumor necrosis factor alpha. Okay, um, and the order Pastorales and the family Pastoralaceae. It also increases the risk of periodontitis, and it is found in both the oral mucosa and the gut. The family Bacteroideales plays a role in periodontitis, and it causes inflammation. So we know that inflammation means if it's causing inflammation in the gums, it's the periodontitis. So it could be a factor. Okay. So these are were the all the microbiota that were causing the periodontitis, and they were increasing the periodontitis risk. These two, that is genus Butyricoccus and genus Clostridium 6, these two decreasing the risk of periodontitis, and they produce a substance that is called the butyrate, which acts as anti-inflammatory mediator. So it helps in inhibiting the inflammation. It also regulates immunity, maintain intestinal epithelial barrier, balance gut microbiota, and it inhibits the cytokines that causes inflammation. So that means they are anti-inflammatory. Okay. So what we conclude that if we target these number of bacteria, and it can help controlling and preventing the periodontitis. So they performed the MR analysis. It was a perfect study, and because it is a suitable study, and it has to find out the causal effects between the gut microbiome and the potential risk factors of periodontitis. And the advantages was that it had no interference of reverse causal effects and confounding factors. It was cost effective as compared to randomized control trial. and it was practical and convincing. But there were some limitations that we will discuss over here. That genome-wide association study was conducted primarily in the European population, um, and the studies can be conducted with other ethnic group as well. The second is they didn't focus on multiple correlations as periodontitis has so many risk factors and there could be so many reasons. And the third limitation is that it was unidirectional role of 196 microbiota because they aimed to explain the risk of periodontitis only. And fourth is they did not explain exact mechanism of gut microbiota, that how do they work and how do they affect periodontitis. So the conclusion is to invent new techniques for the clinical intervention of periodontitis. This article explains the causal relationship between the gut microbiota and the periodontitis, that is inflammation of gums, using two sample Mendelian randomization study. So we can invent the new techniques to control this periodontitis and new treatments. So it's a very good research that was conducted. Okay, so here are the questions that you can see over here, and these are the references. Thank you so much.